Okay, let's learn some stuff. Hi everyone, in the next part of this lecture series, we're going to learn about synapses and also types of graded potentials. So here we go. Well, let's review what we've learned so far. So this picture is showing us a typical neuron, right? We've seen this picture before. And so you can see here toward the left side, you've got the cell body with the dendrites coming in. Right? And we said that graded potentials pass over those areas. Okay? And we learned that graded potentials can vary in strength, and if those graded potentials reach the axon hillock, if the strength of the potential reaches the axon hillock to threshold, then an action potential is going to be triggered. Right? So later on we're going to learn a little bit more about the varying degrees of graded potentials. So when the axon hillock gets stimulated to threshold, then we learn that action potentials are conducted down the membrane of an axon. And this axon, remember, happens to be myelinated. And the type of myelination that's occurring here are from cells called Schwann cells. So that actually tells us this is a neuron from the peripheral nervous system. Okay, and you can see that axons can split. They form collateral axons, and so if an action potential is traveling down the axon, it will also travel down the membranes of those collateral axons, also at the same speed and with the same strength. Okay? And then that brings us to the synaptic terminals at the end. And so remember that here they're called synaptic terminals. They're also known as axon terminals. They're known as boutons. Any of those terms are correct. But then this is where a neuron will either connect to another neuron or another cell, like a muscle cell or an epithelial gland cell or something like that. And so here's what that looks like connecting to another cell. It looks something like this. This is one axon then one neuron connecting to a second neuron. And these neurons then will connect to each other in series. Okay, and so there might be several in a series. But look at the region between the two neurons, that is called the synapse. Okay, so that's where the synaptic terminal of one cell then comes in contact with the dendrite or the cell body of another cell. So the synapse is that region between the two cells. So therefore, here's some more vocabulary for you. Therefore, we can call the cell before the synapse, where the axon terminal or the synaptic terminal is making contact. So the cell before that, we refer to that one as the presynaptic neuron. Presynaptic, because its cell, most of the cell, is before the synaptic region. Okay, the cell that it stimulates, that's after the synaptic region. So that's referred to as the postsynaptic neuron. Okay, so you really want to keep those terms straight, because now you can imagine if our postsynaptic neuron is also connected to another neuron that it can stimulate, then when we're talking about those two neurons, this postsynaptic neuron becomes the presynaptic neuron, and the other one becomes the postsynaptic neuron. Okay, so it just has to do with which cell, whatever synaptic region you're talking about, the cell before it that's doing the stimulating is called the presynaptic neuron, and the cell after it that is getting stimulated is called the postsynaptic neuron. Okay, so a little bit about synapses in general. So I just mentioned this. It's the junction between a presynaptic and a postsynaptic neuron. And there are two types of main types of synapses. And one is an electrical synapse. And that's when they can pass the electrical potential directly from one cell to another. And they can do that because they're actually connected to one another with gap junctions. So if you don't remember gap junctions, go back to the earlier module or earlier page in the module and review the different kinds of connections between cells. So this will be a protein that actually spans the cell membrane between the two cells. So therefore, an electrical potential can pass seamlessly between the two cells. It's much quicker, but their electrical synapses, we believe, are actually pretty rare. However, they are found throughout the nervous system. They have been heavily studied in the brain. But because they're rare, we're going to focus on the second one there, the chemical synapse. So a chemical synapse uses some kind of chemical messenger that gets transmitted from one cell to another. And it has to actually pass a little space between the cells. And so that is what we're talking about happens at the synapse. Okay. So here's another picture showing the presynaptic and postsynaptic cell. Okay, and now you can see the region where the synaptic terminals from the presynaptic cell is coming toward either the dendrite or the cell body of the postsynaptic cell. So if you look down on the left side there in the box, then you can see that box kind of highlighted and blown up for you. So you see there's actually a space between them. I say they come in contact, but really they don't really touch. There's a space between them. And you can see what's happening here with the chemical synapse is the presynaptic cell is releasing a chemical into that space, and that chemical is what's coming in contact with the cell membrane of the postsynaptic cell. Okay. Here's what that looks like under a high-powered microscope. You can see how these presynaptic cell terminals just kind of lay on top of or really close to the cell body of a postsynaptic neuron. And then just to look a little more closely at this, so we can understand the parts of the synaptic region, and then we'll get into exactly how an action potential triggers this functioning at the synapse. So on the right side, that's the postsynaptic cell. And I'm sorry, that's the presynaptic cell. I meant to say presynaptic cell. I know that because inside of it, you see all those red balls in there. Those are actually vesicles that are filled with neurotransmitters. So the neurotransmitter is the chemical that's going to stimulate the postsynaptic cell to the left. Okay, so one of those red vesicles is kind of cut open, and you can see the yellow dots in there. The yellow dots are indicating the neurotransmitter that's stored inside of it. So our neurons make neurotransmitter, and they store them inside of these vesicles. And then when it's triggered, that action potential that's going to come down the membrane of the axon, that's what's going to trigger the release of these vesicles. They're going to merge with the cell membrane and release the neurotransmitter by exocytosis. And the neurotransmitter gets released into the synaptic cleft. Okay, that's the term I wrote there for you. So the synaptic cleft is just that space. It's a fluid-filled space. It's filled with the interstitial fluid, or the ECF, right? Interstitial space, and so it has the fluid in that area. And that's where the neurotransmitter just flows into that space. But they're attracted to the postsynaptic cell, because this would probably be the end of a dendrite, and you can see all those little yellow circles on there, and you can kind of see they're spanning across the cell membrane going into the cell, inside of there, poking through to the cytoplasm. Well, that is a chemically gated channel. Okay, so the neurotransmitter can bind to that chemically gated channel. There's a receptor on the channel. It can bind to it, and the neurotransmitter then triggers the opening of the gate. and that allows an ion to move inside. Okay. Now notice I said an ion moves inside. The neurotransmitter does not move inside of the postsynaptic cell. The neurotransmitter simply binds to the outside of the channel, and that causes a chemical response from that protein of the channel. Remember, the channel is a protein, and it just changes its shape, and it causes the middle to open up to provide an opening. It's specific to certain ions, and in this case we're going to use sodium as an example. And you already learned what happens when sodium enters a cell. You get depolarization. Okay? So it's really important to remember the neurotransmitter does not enter the postsynaptic cell. Okay? It only binds to the receptor on the outside. Okay, and so we'll actually look just a second. We're going to look at, you'll see a little bit how that happens. Okay, looks like the sun is setting. I'm getting sun. My video is going to look funny. Oh well. So here we go. We're going to go step by step and learn how an AP that's coming down the axon triggers the events I just described. So I've got it written for you step by step. So you can see the number I have on the slide corresponds with the number that's on the figure here. So number 1 at the top there, where the axon is beginning to merge into the synaptic terminal, that's the AP, the action potential, coming down the axon along that cell membrane, along the cell membrane of the synaptic terminal. Okay. Now notice in the membrane in the synaptic terminal, those purple things there. Those purple things are also voltage-gated channels, but these channels are specific to calcium. So these are calcium voltage-gated channels. The AP is triggering the calcium channel to open, and when it does, there's calcium on the outside in the interstitial fluid, and so they enter along their concentration gradient. Okay, and that entry of calcium then is what triggers the vesicle to move, and we say it docks along the inside of the synaptic terminal, along the membrane there, just on the other side of the cleft. And that's what you can see at number 3 here. So calcium causes the vesicle to release the neurotransmitter by exocytosis. And so that means the cell membranes are merging, and the neurotransmitter now is flowing into the synaptic cleft. Once it does that, you can see that on the postsynaptic cell, those green structures there, that's the chemically gated channel. And you can see that most of them are closed until one of the neurotransmitters, kind of colored orange there, those small orange balls, the neurotransmitters bind, and that causes the channel to open. Once the channel opens, then you also see the little green balls inside of there. Right? That's sodium. That's a sodium ion. So now it's open. Sodium flows into the cell, right? Into the postsynaptic cell, and that causes depolarization, right? And remember, with a graded potential, it's causing a graded potential inside, because this is at the dendrite or the cell body. And so it's only depolarizing that small localized area of the cell. Okay, so we just got a synaptic response there. Let's zoom in a little bit on those gates, because those are a little bit small to see. And what you can see here now, over on the left side, the first picture there, you can see that it's a chemically gated channel, much like the voltage gated channels we saw when we looked at action potential. They span through the membrane. Okay, but this one, again, voltage doesn't change, open it, it's a chemical. So at the top, You can see that there's sodium flowing around, but they can't enter because nothing is binding to that little site on the left side. That's the receptor. Okay, and that receptor is also specific. It can only certain neurotransmitters can bind to it. Usually it's only one. Sometimes other drugs and things can bind to that. So we'll talk about how drugs can influence our nerves because they can also bind to some of these receptor sites. Okay, in the middle panel, you see that the neurotransmitter being used, now we're using the name of one, ACH, that's called acetylcholine. I'll go over those later. ACH can bind, and when it does, it opens that channel. Okay, so now it's open and sodium can flow through. And sodium is flowing in along its concentration gradient. But also remember the inside of the cell is negative, so it is also flowing in based on its electric gradient as well. Okay. Once the neurotransmitter is removed, you can see that in the last panel, the gate closes. And once that closes, the sodium can no longer flow through. So you can see we have a little bit of a control system going on here, how we can control the opening and closing of those gates, and therefore we can change the strength of the signal. Okay, you can imagine either the longer those gates are open, or if more of those gates are open, then more sodium can flow through. Alrighty. So that's part one of this lecture. Next, we'll actually look how the stimulation of a postsynaptic cell can vary in strength or even inhibit the depolarization of the cell.